Hi there, this video is about CD8 cells, what they are and the role they play within the immune system. Undifferentiated lymphocytes are transported from the bone marrow to the thymus, presenting both CD8 and CD4 glycoproteins. In the thymus, the CD8 cells are exposed to self-antigens bound to MHC and they undergo selection. In positive selection, the binding is too weak, and in negative selection, the binding is too strong, which could lead to autoimmunity. Therefore, in both, they are subject to apoptosis. The cells which survive selection differentiate into CD4 or CD8 T cells.
Naive CD8 cells, fresh from the thymus, need to be activated by simultaneous signals. These signals are antigenic stimulation through the CD8 cell receptor and antigens presented on MHC1, co-stimulation through molecules CD80 and 86 binding to CD28 stop anergy of the CD8 cells, and stimulation by inflammatory cytokines like interleukin-2 and interferon alpha, which helps the CD8 cell become cytolytic. Mature dendritic cells recruit CD4 helper T cells and activate CD8 cells, enabling mass killing of infected cells by each CD8 cell, known as serial killing. Once activated, they clonally expand with help from interleukin-2, increasing the number of specific CD8 cells, which then migrate to all tissues of the body.
The purpose of CD8 cells is tumour surveillance and to protect against intracellular pathogens like viruses and bacteria. The only option to prevent further spread of the pathogen or cloning of the tumour-causing mutation is the destruction of host cells that are involved. Cytotoxic T cells have three mechanisms of killing. Firstly, they release perforin and granzymes in a directed fashion. Perforin produces a pore in the membrane of the targeted cell in a manner similar to the membrane attack complex in complement. The pore allows granzymes to enter the target cell and cleave proteins inside the cell, initiating apoptotic signaling pathways. Secondly, they secrete cytokines like TNF-alpha and IFN-gamma, which have anti-tumour and antiviral effects. The final mechanism is by Fas-cell protein interactions, which leads to a cascade and apoptosis. At the end of the immune response, 5 to 10% of the CD8 cells are converted into memory CD8 cells, which are long-lived and circulate at potential sites of entry.
CD8 cells have a role in anti-tumour surveillance, which protect against oral cancer. The dentist should make note of a patient who is immunodeficient in any way, as this leads to an increased risk of oral cancer. Patients with a lower T cell count might develop oral candidiasis, as well as increasing the susceptibility to major allergic responses. CD8 cells inhibit IgE, which limit allergies. A decrease in these cells means an increase in IgE. The CD8 count is also useful when following the progression of HIV. This could be important data for the dentist to have when treating the patient, as they can look for any oral changes as the disease progresses. As well as this, herpes affects CD8 cells by downregulating the MHC1 of the cell it has infected. In this way, the virus can successfully hide from the CD8 cells, and they struggle to eliminate the virus. As a dentist will see many patients with herpes simplex virus, it is important to bear in mind.
Overall, CD8 cells play a big role in the protection against intracellular diseases, many of which find entry through the oral cavity. As a dentist, it is vital to understand about immunology so that the best treatment and explanations can be given to the patients. Thank you for listening.